So today we will learn the chapter 11. Chapter 11 title is Protein Folding in Vivo and in vitro. Chapter 11 is composed of three subjects starting from the factors determining the protein fold, and secondly the amide exchange and the measurement of protein folding. Finally, we will cover kinetic barrier to refolding. Okay, so in this slide, I will explain the first subject, factors determining the protein fold. So first of all, I will explain two important terminology, denaturation and folding. So denaturation means that a process in which proteins lose their shape and function. Okay, so this is protein in native structure. Okay, so denaturation means that a process in which proteins lose their shape and finally they will lose their function. So this is the denaturation. Okay, so this is the graph, a typical thermal or chemical denaturation curve over two-state protein. One state is native, the other state is denatured. Okay, so x-axis is perturbant. Okay, so protein can be denatured by perturbant. Perturbant can be the chemical denaturant or heat to disrupt the weak bond stabilizing tertiary structure. Okay, so the perturbant can be increasing concentration of denaturant or increasing temperature. Okay, so y-axis means that the status, so y-axis 0 means that the fully native status, and FD1 means that fully denatured structures. Okay, so when the denaturant concentration was increased or when the temperature was increased, okay, so native structure and become the denatured structure. Okay, so what is folding? Folding means that a process by which linear proteins form their unfolded structure. So linear proteins form their unfolded structure. So this is we call it as folding. Okay, a process, once again, a process by which linear proteins form their unfolded structure, we call it as folding. So in this slide, I will explain the first factor determining the protein fold. The first factor is denaturant. So previously we learned about chaotrops. So chaotrops belong to the denaturant. So this is a denaturant. Okay. The first of all, denaturant disrupt hydrogen bonding between water molecules. So denaturant disrupt hydrogen bonding between water molecules, resulting in the decrease of the hydrophobic effect within the solution. So when the hydrophobic effect exists, so usually it can make the hydration cell. So this is the water. So it can make the hydration cell like this. Okay, so it means that decrease in hydrophobic effect around the hydrophobic amino acid cannot make the hydration shell around the proteins. So it means that it can decrease the hydration shell making. So maybe resulting in the formation of the denatured protein is like this. Okay, so secondly, denaturant can disrupt some of non-covalent interaction that stabilize protein tertiary structure. Okay, in the previously chapter 3, we learned the interactions stabilizing tertiary structure, starting from the covalent bonding and also ionic, and finally we also mentioned about van der Waals interaction. So denaturant can disrupt non-covalent interaction. So they can disrupt non-covalent interaction starting from the ionic, hydrogen, hydrophobic, van der Waals interaction. Okay, and also guanidium and the urea is belong to the denaturant. Okay, previously we also learned that stabilizing property of cosmotropes and chaotropes. At that time we learned that the very famous chaotropes is guanidium. Okay, so guanidium is the this is guanidium structure. So they have the at the center they have carbon. After that they have the amine group, amine group, amine group. So this is we call it as guanidium cation. Okay, so guanidium has positively charges. Okay, so it can disrupt non-covalent interaction. And also another denaturant is urea. This is urea structure. So they are nearly similar to the guanidium cation. At the center they have the carbon. They have amine group and amine group. So instead of this amine group, they have the carbonyl group like this. Okay, so but the function is nearly similar to guanidium. Okay, so urea is they are neutral, but this one also can disrupt non-covalent interactions. Okay, so in this slide, I will explain the second factors determining the protein fold. So we call it as a hydrophobic effect. So previously in chapter 3 and chapter 9, we learned about hydrophobic effect. So what is hydrophobic effect? So hydrophobic effect means that in the presence of water, so hydrophobic amino acid is clumps together. So this is we call it as the hydrophobic effect. And also when we learn the cosmotropes, so this is cosmotropes ion. Okay, so cosmotropes ion exhibit usually the strong interaction with the water molecules. So cosmotropes exhibit the strong interaction with water molecule, so resulting in the increase of the hydrophobic effect within the solution. So increase the hydrophobic effect within the solution. So it means that cosmotropes ion cause proteins to precipitate in proteins' native structure. Okay, so once again, the hydrophobic effect is a major driving force for protein folding. And also it can induce the barrier of the hydrophobic residue in the center of the protein complex. For example, this is a polypeptide. Okay, so this is the hydrophobic residue amino acid. So this is the hydrophobic amino acid. It's like this, and this is water. Okay, so hydrophobic effect induces the barrier of the hydrophobic residue in the center of a protein complex. So, okay, once again, second factor determining the protein folding is hydrophobic effect. This effect is major driving force for protein folding, and also it can induce the barrier of the hydrophobic residue in the center of a protein complex. So in this slide, I will explain the third factors determining the protein fold. So we call it as accessible space area. Okay, so accessible space area increased as proteins is denatured. So this is a native protein which is stabilized by covalent bonding and non-covalent bonding. So when denaturation happens, so denaturant disrupt non-covalent bonding. So this is the denatured proteins. So if you see here, the accessible area, so accessible area is increased as the protein is denatured. Okay, so this is also graph to show that x-axis is the concentration of the perturbant, and the y-axis means that degree of the denaturation. So FD0 means that the protein is in native structure, and FD1 means that protein is fully denatured. Okay, so when as the concentration of denaturant is increased, it means that protein can be fully denatured. So as a protein is fully denatured, the accessible space area is also increased. And also accessible space area is increased. This is the side chain of the non-polar amino acid, starting from the alanine, threonine, the leucine, methionine, phenylalanine, tyrosine, and tryptophan. So if you see the size of the non-polar side chain, the side chain is the alanine is composed of the methyl, and this is the threonine, the leucine, and the methionine. Okay, so and also phenylalanine is also composed of the methyl, and also they are phenyl. Phenyl is composed of 6 carbon, so this size is larger than this one. And also tyrosine is also larger than phenylalanine, and tryptophan is the largest amino acid. Okay, so also you can find that accessible space area is increased as the size of non-polar side chain is increased. Okay, so based on this observation, accessible space area can be used as a factor that determines the degree of protein fold. So once again, the accessible space area can be used as a factor that determines the degree of protein fold. Okay, so when the accessible space area is the higher, means that the protein is denatured. When the protein space area is very low, it means that protein is in native, when protein is in the native status. Okay, native or folded structures. Okay. In this slide, I will explain the free energy changes during protein folding from unfolded to folded status. Okay, so this is the free energy in the unfolded proteins. The free energy of the unfolded protein looks like this. Okay, also this is the free energy in folded proteins. They are very low, is like this. So it means that free energies decrease during the protein folding from unfolded to folded. So it means that delta G is the free energy change. Okay, so G free energy unfolded proteins free energy minus folded proteins free energy. Okay, so delta G is the negative. Okay, so if you see here the definition of the free energy change of reaction, so if delta G is negative, means that loss of the energy, and means that reaction goes spontaneously. Okay, so it means that reaction from unfolded to folded status goes spontaneously. Okay, so this is the thermodynamically favorable process. Okay, so in this slide, I will explain the new terminology, conformational stability, delta G. So conformation can be either unfolded or folded status. So previously in chapter 3, we learned that the interaction stabilizing tertiary structure. So interaction is composed of the covalent bonding and also non-covalent bonding, which consists of the ionic, hydrogen bonding, and hydrophobic effect finally there is the van der Waals interaction. So folded proteins can be changed to unfolded protein when the non-covalent bond is disrupted. Okay, so the conformational stability means that in the absence of denaturants, if there is no denaturant, so free energy changes during the protein folding from the unfolded conformation status to folded conformation status. So it means that delta G is the free energy at unfolded status minus free energy at folded status. So this is delta G conformational stability. Important thing is in the absence of denaturant. So in this slide, we will learn how to calculate conformational stability, delta G. Okay, so previous slide we learned that delta G is that free energy at unfolded proteins minus free energy at folded proteins. Okay, so now I will show you the easiest way to calculate conformational stability. Okay, so U means that unfolded protein, and F means that folded proteins. So in the absence of denaturants, unfolded protein can be easily changed to folded protein, and also folded protein can be changed to unfolded protein. So, and also the reaction constant from the unfolded protein to folded protein is recorded as K1, and also reaction constant from the folded protein to unfolded protein recorded as K2. And also that's why the equilibrium constant KEQ can be the K1 divided by K2. And also the KEQ KEQ can be expressed as the concentration of the folded protein divided by concentration of the unfolded proteins. So, okay, so delta G can be RT. R means that universal gas constant. So R is the value is 8.314, and T means that Kelvin temperature. So T is the 273 plus Celsius temperature. And also Ln means that natural log, and KEQ means that comes from the equilibrium constant. Okay, so at the middle point of the unfolding transition between the folded and denatured status. So between the folded and denatured status, at the point is that the middle point. So what is the equilibrium constant? So KEQ means that the concentration of folded proteins divided by concentration of the unfolded proteins. So it means that because the reaction is at the middle point, so it means that the concentration of folded protein can be 0.5, and concentration of unfolded protein, we also call it as 0.5. So it will be KEQ will be 1. It's like this. Okay, so in this slide, I will show you one example how to calculate conformational stability. The question is that the result of the experiment revealed that the rate of unfolding in the absence of denaturant. So in the absence of denaturant, the rate of unfolding. The rate of unfolding means that K2. So K2 will be 10, while the rate of refolding means that K1, the rate of refolding is that 2 multiply 10, 5 factorial. Okay, so it means that the K1 is like this and K2 is like this. So KEQ means that K1 divided by K2. So K1 is the 2 multiply 10 5 factorial should be divided by 10. So it will be the KEQ value. Okay, so the process is two-state and is the best estimate of conformational stability for this reaction. So at that time, the temperature is room temperature. Okay, so this is delta G. So we have to calculate conformational stability. So conformational stability is that delta G is that RT and natural log KEQ. So R means that 8.314 and T means that 273 plus room temperature is, we assume that this is 25 centigrade, and also multiply natural log, and KEQ is that 2 multiply 10 4 factorial. So it means that delta G will be 24,000 joule mole. It's like this. So we can change it like this. This value is approximately 25 kilojoule per mole. So the conformational stability is around 25 kilojoule per mole. Okay, so in this slide, I will show you a new concept, conformational stability in the presence of denaturant. So previously we learned that conformational stability in the absence of the denaturants. Now in this slide, we will learn how to calculate conformational stability in the presence of denaturant. So we call it as a delta GU. Okay, so delta GU is equal delta GU H2O minus MEQ multiply D. Okay, what is delta GU H2O? So delta GU H2O means that conformational stability in the absence of denaturant. Sometimes we just write down like this, delta G H2O. So this is the same meaning. Okay, and also this figure 11.9 show that 1, 2, 3, 4. 4 ideal denaturation curve representing a protein with a conformational stability. So in the absence of denaturant, delta G H2O was the 20 kilojoule per mole. Okay, so the value in the absence of denaturant, conformational stability value was the 20 kilojoule per mole. And also the curve reflect the different value of MEQ, ranging from the red is that MEQ 10,000, and blue means that MEQ 8,000, and green means that MEQ 6,000. Okay, so this graph is to show that x-axis is that denaturant concentration. So y-axis means that fraction folded. 1 means that all of a protein is folded, and 0 means that all of a protein is unfolded. Okay, so when the half of the protein is folded or half of them protein is unfolded, okay, for example, in this red curve, okay, the concentration of denaturant concentration to make half of them is the folded and half of them is unfolded is 2. How about when the MEQ is 8,000, the denaturant concentration is around the denaturant concentration to make the half of them is folded and half of them is unfolded is around 3.3. How about when the MEQ is 6,000? the denaturant concentration is around 5. Okay, so it means that if the value is higher, it means that the binding between the denaturant and protein is higher. Okay, that's why at lower concentration, for example, in this case, at lower concentration, half of them is folded and half of them is unfolded. However, when the MEQ is 6,000, around 5 denaturant concentration, half of them is the folded and half of them is unfolded, because their binding constant is very low compared to the MEQ 10,000. And also the D means that molar concentration of denaturant. Okay, so this is so D means that molar concentration of the denaturant. Okay, so we will see some details of how to calculate delta GU in the next slide. Okay. So in this slide, I will explain the details procedure to calculate conformational stability in the presence of denaturant. So previously we found that delta G H2O was 20 kJ per mole. It means that y-intercept will be the 20. So it means that delta GU will be the 20 minus MEQ and D. So D means that molar concentration of denaturant. Okay, so x-axis is the denaturant concentration. So we can change this graph is y is 20 minus MEQ x is like this. Okay, so red color curve, the MEQ was 10,000, and the blue color curve was MEQ was the 8,000, and the green color was MEQ was 6,000. So we can change it. The graph is like this. So for example, the red color curve, we can show it like this. Y is 20 minus 10,000 x, and the blue curve means that y is 20 minus 8,000 x, and green curve will show that y is 20 minus 6,000 x. Okay, so what's the meaning of this graph? Okay, for example, the blue curve, so MEQ was 10,000. It means that denaturant can strongly bind to the proteins. Okay, so it means that the conformational stability is decreased very quickly, like this. So how about when the MEQ was 8,000? So the conformational stability is decreased not very quickly, it's like this. Okay, so how about when the MEQ was 6,000? So it means that the binding capacity of a denaturant to protein is very low. It means that the conformational stability is decreased very slowly. It's like this. Okay, so once again, the conformational stability in the presence of denaturant is recorded as a delta GU. So delta GU is delta GU H2O. So delta GU H2O means that conformational stability in the absence of denaturant, and minus MEQ means that binding constant between the denaturant and protein, and D is the molar concentration of denaturant. Okay, so in this slide, I will explain the final factors determining the protein fold. So we call it as a temperature. So temperature is the final factors determining the protein fold. So previously we learned that interaction stabilizing tertiary structure. starting from the covalent bonding, and also there is the non-covalent bonding, which is composed of ionic, hydrogen bonding, and hydrophobic effect. Finally, there was the van der Waals interaction. But the temperature can function as a perturbant, and temperature can only disrupt non-covalent bonding starting from the ionic and finally van der Waals interaction. So also, the temperature is a major driving force for protein unfolding. So this is folded protein, and so when we increase the temperature, proteins become unfolded. So it means that protein can be unfolded by increasing temperature. Okay, so this is also graph to show that x-axis is perturbant, especially the temperature, and the y-axis is the fraction unfolded proteins. So FD0 means that all of protein is folded, and FD1 means that all of protein is unfolded. So if we increase the temperature, so folded protein can be changed to unfolded proteins. So once again, the temperature is the major driving force for protein unfolding. So it means that temperature can be the final factors determining the protein fold. Okay, so in this slide we will learn the thermal stability curve. Thermal stability curve is the fractionation of native proteins or fractionation of the folded proteins as a function of temperature. It means that y-axis is the fractionation of native proteins or fractionation of folded protein, and x-axis is temperature. Okay, so in y-axis, number 1 means that all of protein is existed as the folded structure, and number 0 it means that all of protein existed as a denatured or unfolded structure. Okay, so thermal stability curve enable us to measure the protein stability. So let me show you one example. Okay, number 1 is the control protein. Okay, but number 2, 3, 4, so we made some mutation. Okay, so this is the destabilized variant. Okay, so this is the destabilized variant proteins. Okay, so after that we measure the we measure the fractionation of native protein percentage. Okay, so this is the number 1. Okay, this is number 2, number 3, and number 4. So when this means that the fractionation native protein is 0.5, means that half of them existed as the folded protein, and half of them existed as unfolded or denatured structure. Okay, so let me show you. Let me see the number 1. So number 1 means that the temperature is around 78. So number 1 means that at 78 centigrade, half of them is existed as native structure, and half of them is existed as the unfolded structure. How about number 2? Number 2 is half of them exist around at 70 centigrade, half of them is existed as native, and half of them is existed as unfolded protein. How about number 3? Number 3 is around at 60 centigrade, half of them is existed native, and half of them existed unfolded protein. How about number 4? So number 4 is that half of them at around 45, half of them is existed as native, and half of them is existed as the unfolded protein or denatured proteins. Okay, so it means that how about the number 1? At 45, all of number 1 is existed as the native structure. So it means that number 1 is more stable than number 4. Okay, so let me show you one example. So it means that based on this graph, we can say that number 1 is more stable than number 2. Number 2 is more stable than number 3. And number 3 is more stable than number 4. So based on this thermal stability curve, we can tell us which protein is more stable or not. So okay, based on thermal stability curve, okay, so we can put the graph, we can put the line at 0.5. So at that we can measure the temperature. So based on this temperature, we can say that which protein is more stable, which protein is more stable, which protein is more stable, and finally this is the weakest protein. Okay, so in this slide we will learn the differential scanning calorimetry. So we call it as DSC. So DSC can measure the energy absorbed or energy released by a sample as the sample is heated. Okay, so this is one example of DSC. Okay, so one container we put the sample. Sample is composed of the protein and solvent. Another container we put we call it as a reference. So we only put the solvent. Okay, so after that we can measure the energy is absorbed or energy is released by a sample, okay, as it is heated. Okay, by using this machine, we can measure delta Cp and the Tm. Okay, so this is the graph. The x-axis is time and the y-axis is heat flow. Okay, so this is at the starting time. Okay, so when the sample is the absorbed energy, it means that the graph will be decreased because the heat flow will be because the sample will absorb the energy. When the sample is release the energy, so maybe the graph will be go upside is like this, go upward like this. Okay, so this is the baseline. So as the sample is heated at certain time point, so heat flow will be decreased. So it means that the heat capacity is increased. Okay, and also as time goes by, we find that at certain point delta Cp is the highest, is like this. Okay, so this is, we call it as TM, transition midpoint temperature. Okay, and delta Cp means that change in heat capacity because they absorbed the energy. So it means that delta Cp is increased, and also at Tm point delta Cp is highest, is like this. Okay, so by using DSC, so we can measure the heat flow as a function of time. So now we can change this graph into like this. Okay, so this graph is the x-axis is temperature and the y-axis is the heat capacity change, delta Cp. Okay, so in this graph we have the two graphs. One is the delta Cp changes in the solvent. One is the delta Cp change in the sample. Okay, for example, for solvents, so as the temperatures increased, solvent absorb the energy. So it means that delta Cp is increased very slowly like this. However, in the sample, sample is consist of protein and solvent. So the delta Cp is increases like this, but at certain time point delta Cp increased abruptly. So that delta Cp increased abruptly. After that, the delta Cp is decreased abruptly like this. Okay, so we have the two graphs like this. So now by using these two graphs, we can make the corrected calorimetry profiles. So we can make the graph is like this. Okay, so this is the corrected calorimetry profiles like this. So it means that this is the Tm. So this is Tm. Tm means that when delta Cp is highest, we call it as the transition midpoint temperature. Okay, so before the Tm, we can predict that protein has folded structure, but after Tm, we can predict it protein has unfolded structure. It's like this. Okay, so by using the DSC data, so we can measure the Tm. So based on Tm, we can predict that protein has a folded structure while protein has the unfolded structure. So in this slide, we will learn the second subject, amide exchange and the measurement of protein folding. The main principle of this experiment is that hydrogen in the amide group involves in the hydrogen bonding. So what is amide group? Okay, so this is the peptide bond between the carboxyl group and the amine group. So we call it this group as the amide group. Especially the hydrogen in the amide group is involved in hydrogen bonding. So let me show you one example. So previously we learned secondary structure of alpha helix. So alpha helix is stabilized by the hydrogen bonding between the hydrogen in the amide group and also the neighboring oxygen. So this is the hydrogen bonding. Okay, so in the hydrogen bonding, so the hydrogen in amide group is involved in the hydrogen bonding. So we also have to know to fully understand this experiment, we have to know the isotype of hydrogen. So this is the usual hydrogen, which is composed of the one proton and one electron. That's why we call it as a H, and also their molecular mass is 1. And however, there is another isotype is available. So they have the proton. In addition to proton, they have a neutron. And that's why we call it as a deuterium, and we write down D, and the molecular mass is 1, 2. So write down here 2. Okay, the procedure of this experiment is, first of all, dissolve the proteins in a D2O containing 6 mole guanidinium. First of all, dissolve proteins in D2O containing 6 mole guanidinium. So it means that 6 mole guanidinium solution, okay, can denature the proteins. So protein can be denatured. So also the solution has the D2O. Okay, so proteins can be labeled with deuterium. So during the denaturing step, protein can be labeled with deuterium. Okay, so this is the first step. Okay, so after that, so this is the denatured protein which is labeled with deuterium. So after that, place proteins in the D2O. It means that protein can initiate the refolding. Especially at this location, hydrogen bonding happens between the deuterium and the nearby oxygen and deuterium and nearby oxygen. So after that, we can place the proteins in the H2O, especially at high pH, to remove the deuterium from proteins. So I will show you the principle of this experiment. So this is the amino acid which is located at the neutral pH. So this is the amino group and this is carboxyl group, and carboxyl group has the proton like this at neutral pH. So when we put this amino acid at high pH, it means that the high pH means that the H plus level is very low. So it means that this H, which is located at the carboxyl group, can be added into the solution. So it means that this H can be removed. Okay. So once again, so we can place the proteins in H2O at high pH to remove deuterium from the proteins. So it means that this deuterium is not involved in the hydrogen bonding. So this deuterium can be removed like this. Okay, can be removed like this. Okay. Okay, so after that, place proteins in H2O, not D2O. Place protein in H2O at the low pH to add H in the proteins. So I will show you the principle of this experiment. Okay, so this is the amino acid at neutral pH. So when we put this amino acid in at the low pH, means that at low pH solution has excess protons like this H plus. So it means that excess H plus can be easily added into the amino acid is like this. So when we place this protein in the H2O, especially at the low pH, so it means that H1 can be added like this, added to the proteins like this. So this is the third step. And so finally, we can do the NMR analysis of proteins. So this machine to show that NMR spectrometry instrument, so they use the magnet. But I will not explain the principle of NMR, but I will explain that NMR can measure the exact concentration of the deuterium. Okay, so the higher concentration of deuterium indicates that protein has a folded structure like this, and the lower concentration of deuterium indicates that protein has unfolded structures like this. So through the amide exchange method, we can measure the degree of the protein folding. So whether the protein is highly folded or protein is unfolded. Okay, so this is the amide exchange, and we can measure the protein folding degrees. Okay, so in this slide, we will learn the third subject, kinetic barrier to refolding. So figure 11.25 show that on and off pathway intermediates during protein folding. Okay, so this is the U is unfolded protein and F is folded proteins. So between U and F, so there is I1 and I2 is available. So because I1 and I2, we call it as the on pathway intermediate. Okay. However, there is another possibility. For example, X1 or X2 can be available. Okay. So X1 and X2, we call it as the off pathway intermediate. Okay. So when the off pathway intermediate is present, okay, so usually the standard reaction starting from the unfolded I2, I1, I2, F. However, when the off pathway intermediate is present, U can go to the X1, and X1 can go to the U, and U can go to the I1, and I1 can go to the X2, or X2 can go to the I1, and I1 can go to the I2, and the I2 can go to the forward. So it means that reaction rate is very slow, and also when off pathway is present, unwanted folding occurs. So usually the folded protein should be like this, but however, when off pathway intermediate is present, unwanted, for example, the more fully folded or is not loosely folded. So it means that this is unwanted folding can occur. So in this slide, we will learn the example of off pathway intermediate. Okay, so this is the one amino acid, and this is another amino acid. Between the amino acid, there is peptide bond is available. So this amino acid, we call it as X amino acid, and this is proline. They have ring, and this is proline. They have a ring. It's like this. Okay, so cis trans isomerization of peptide bonds preceding the proline residue. Okay, so this is a cis form. For example, this is a C alpha protein, and this is also C alpha proteins. So when the C alpha protein is located in the same location, so we call it as this configuration is cis form. However, this is another configuration. C alpha is located here, and C alpha is located the other location. And C alpha is located here, and C alpha is located another position. So we call it as this is a trans configuration. Okay, so cis and trans isomerization of peptide bonds preceding proline residue is available, like this. And also the ratio of the cis and trans configuration in XAA to the proline is the 1:4. Most of the form is existed as a transform. Okay, so previously we learned that in chapter 3, we learned that the primary structure, when the proline is located here, so it makes the turn is like this. So this is the trans configuration. Okay, so cis configuration in XAA and proline. So this is a cis configuration. Okay, so this cis configuration in XAA proline can be a kinetic barrier to refolding, or cis configuration in XAA proline can be off pathway intermediate. This can be off pathway intermediate, and also this configuration in XAA proline can be a kinetic barrier to refolding. So for example, unfolded protein can go to the folded status, but unfolded is go to the intermediate. After that, intermediate I1, and after that, it can change it to cis configuration. So okay, after that, it can change it to I2 and F. Or sometimes unfolded protein can go to the X1. This can be the cis configuration in the XAA and proline. Okay, so once again, the example of the off pathway intermediate is the cis configuration in XAA proline. So it existed 1:4 ratio. So 1 is the cis configuration in XAA proline. So this can be the kinetic barrier, or can be off pathway intermediate to refolding. Today we learned the chapter 11. The title was Protein Folding in Vivo and in Vitro. So in chapter 11 we covered 3 subjects. starting from the factors determining the protein fold, we learned the 4-factor, and also we learned the amide exchange and the measurement of protein folding. So finally we covered kinetic barrier to refolding. Okay, so today we will finish class